Okay, so this is our chapter 10 and 11 study points. So basically, the reason why we study DNA, specifically bacterial DNA, is because of genetic variation. Genetic variation is the reason why we have mutations, why we have different species, and why we have different strains of bacteria. Proteins and bacterial DNA replication also acts as really good targets for antibiotics. The more we know about DNA replication and its proteins, the more we can make new antibiotics to fight off emerging bacteria. Explain the flow of genetic information. This is just what you need to know, just like the order and steps. The DNA, the transcription, RNA, the translation of protein. You do not need to know the specific details. Describe genotype. So basically, a genotype, I think about genetic makeup as the same, like beginning of the word, genotype, genetic makeup. And basically, it's just a blueprint of and information of all organisms. Phenotype, I think about physical traits, properties of the genotype that are observable, right? So like, what's it called? Like what it looks like, if it's like round or if it's like sticky, stuff like that. And it basically can be influenced by environment. Explain the ways that genotypic changes occur in bacteria. So basically, it happens in two ways. It's either mutations or gene transfer. So mutation, it happens in a cell alone, due to like DNA sequences and like it being changed or being done incorrectly. While gene transfer is basically where we like a cell takes new genetic information from donors and are basically like they get new traits from that, if that makes sense. So it's from cell to cell, not just on its own. So vertical versus horizontal gene transfer, right? So vertical is where it's up and down, right? Vertical is up and down, while horizontal is side to side. So up and down as in mother to daughter. So it's gonna be genes that are shared from generation to generation. Horizontal is where it's shared between cells of the same generation. Just note that, that's really important. Explain what plasmids are. Know the features of plasmids. So plasmids are non-essential, but they are very helpful in coding for genetic traits. All right, and not all bacterial cells have plasmids, but if they do, they're more what's it called? They're more effective, and they live longer, and they are self-replicating, and they have a narrow host range, and I'll get into that later. What is an R plasmid? So resistance plasmid, right? So resistance plasmid is where it is transmitted via conjugation or transformation, which is one of our forms of gene transfer, and also replicate independently of host. However, it has a wider host range, unlike plasmids. And what you really need to know is that it codes for resistance specifically against beta-lactamase drugs. For example, if you're given a beta-lactamase drug for like a bacterial infection, but you don't get better, it's probably likely that you have a R plasmid in your bacterial cells, which is why it's not working. What kind of genes might be carried on plasmids? So our plasmid encoded traits are antibiotic resistance, paleosynthesis, which is a form of this is how like gene transfer is conducted through one of our forms of gene transfer, which I'll get into later. Toxin production, so it makes the bacteria more toxic and more deadly. Capsules and biofilms just aid in defense of the bacterial cell. What must plasmids have in order to be transferable? So like I said, so regular plasmids, not resistant plasmids, regular plasmids have a narrow host range, right? So they need to be transferred with another bacterial cell that is double-stranded and also has similar characteristics to the recipient, or to the host, sorry. Explain the 3 different mechanisms of gene transfer. So transformation, transduction, and conjugation are 3 forms of gene transfer. And basically, transformation is where it takes up DNA directly from the environment. Transduction is where bacteriophage, remember lysogenic conversion. Transduction is where bacteriophage injects a hybrid of viral DNA and DNA into other infected bacterial cells, right? So basically, you need to know it injects. Conjugation, it's transferred. So DNA is transferred between cells via conjugation Pillis bridge, which I mentioned earlier. And basically, only this form of gene transfer is only between 2 living cells. So transformation takes up DNA. Transduction injects a hybrid. Conjugation is transferred. DNA is transferred. Okay, so explain the experiment that demonstrated gene transfer in bacteria. So this is Griffith's experiment with mouse. So basically what he did was that he injected mice with living encapsulated cell. And when that happened, the mouse died. When injected with a living non-encapsulated cell, nothing happened. But when he injected heat-killed encapsulated cells, so the previously living encapsulated cells was killed with heat, nothing happened, right? But then he was like, let me combine the 2 with the living non-encapsulated cell and the heat encapsulated cell where nothing happened, combine the 2 and see what happens to the mice. So the mouse actually died. And basically what this proves to us is that the living non-encapsulated cell was able to conduct gene transfer. Why? Because it picked up genetic material, which is transformation, right? Picked up genetic material from the heat kill cells and became pathogenic. And so basically it became a living encapsulated cell again. So describe and understand the steps of Griffith's experiment. So basically you begin with 2 plates of GSA, and these plates can only synthesize if it's given the right amino acids. So what happens is this side is that it's only half of the amino acids it needs, and this side is only the other half of what the GSA needs to grow, right? So when plated, they were only given half, so they could not grow. However, if you mix the 4 amino acids together, then it shows that it is able to grow finally. Basically, it just shows that bacteria share genetic information in order to grow colonies. Explain the cholera toxin example of transduction. So basically, a patient is diagnosed with cholera and they're prescribed doxycycline, but it does not work. The reason why it did not work is that the cholera bacteria, it has found to undergone lysogenic conversion, right? And now it carries cholera toxin gene due to horizontal gene transfer, and that is specifically transduction. Remember, because like I said, we're getting that bacteriophage and we're injecting viral DNA into bacteria cells. Explain the staphylococcus case study. So basically, a patient is diagnosed with necrotizing fasciitis. They're put on an IV broad spectrum antibiotic, but the patient seems to get worse. The patient contracted a strain of MRSA, and it's basically a Staphylococcus aureus strain that is methicillin resistant. And the strain also had toxins that the strain of MRSA had toxins that limited blood flow and restricted circulation of IV antibiotics. So basically, it had these like superpowers that basically stopped like the antibiotics to be like delivered to the rest of the body. This is why the patient died. And due to horizontal transfer of prophage between strains of Staphylococcus aureus. So which horizontal transfer method is that? That is going to be, I think, transformation, transduction, conjugation, because it's between strains of Staphylococcus aureus, living strains. Explain the Bacillus anthracis case study. So basically, patient tans leather hides in Africa, and the patient gets sick and is suspected to have anthrax. They're medicated with IV penicillin but died, and the patient likely inhaled spores because when you like endospores, because when you tan hides, endospores are released, and then the patient inhaled it, got sick, and died. And the reason why the IV penicillin did not work is because specifically Bacillus anthracis is very, 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 very toxic. These toxins are really bad. And these two plasmids that carry the toxin gene basically makes the bacteria more virulent and pathogenic. And basically, low-key, the patient had no chance of survival. Briefly describe the different types of mutations. So these are our types, like different terms really, but you need to know specifically spontaneous mutation and induced mutation, and I'll go over that in the next one. But basically, non-mutated cells are called wild type, mutants are called mutated cells, and the rest I don't think you really need to know. But basically the rest are just like changes in the genetic like a mistake in the genetic code. And distinguish between spontaneous and induced. Spontaneous is basically a mutation and error in DNA replication, and it happens out of nowhere. It's just you're replicating your DNA and then you made a mistake. However, induced, you have to think about outside factors. So basically the DNA is damaged by like radiation exposure, right? Radiation kills DNA, so therefore the DNA is damaged and it becomes mutant. Explain the experiment for selecting for mutation. So basically, you have this vial of sensitive cells and resistant mutant cells. So when you pour the vial onto a plate, like an agar plate, with an antibiotic that can kill these sensitive cells, basically the antibiotic will kill sensitive cells, and then the ones that are not killed are considered mutants and are resistant to the antibiotic. What are bacteria mutagenesis assays for? So basically, it screens bacteria for potential carcinogens, which are mutants that are very or could be harmful to the human body. And basically, we need to know these because, we know what is harmful. Like, for example, like if this chemical is harmful to us or not. But by doing these mutagenesis assays, they're less expensive, extensive, and more simple than testing on animals. And this does come up on the test. Explain the Ames assay. So it's one of our mutagenesis assays, and basically same thing. It screens bacteria for potential carcinogens, but specifically, if like colonies grow on your Ames assay, then that means that a mutagen is present. Because, for example, like you're just like pouring histidine to see anyways, you don't really need to know, but if colonies grow, a mutagen is present on the Ames assay. And that's it.